Okay, this is the second of the Miami University students' talks. So hi, I'm Lauren, if you don't know who I am. I'm going to be talking about the research that I did with the taxonomy of the Trifolium Gracilentum species complex. Okay, first starting with a little bit of context, clovers are in the Fabaceae family or the legume family, which is the third largest family of flowering plants. It's very economically important. Lots of these species are grown as crops, and it's also important to the environment since lots of them have associations with nitrogen-fixing bacteria. Then within this family, the Trifolium genus contains all the clover species. There are currently about 250 different species of clovers recognized. Many of these are cultivated. There's at least 16 species of clovers that are cultivated. Some of the most common being the white clover at the top of the slide there and the red clover at the bottom. They're cultivated for various purposes, some of the big ones being livestock forage crops or green manure crops, and because of this, they've been introduced to many places around the world. But even before their introductions, clovers were pretty widespread overall, with three main centers of diversity in the Mediterranean, Western North America, and Eastern Africa. And the group that I'll be focusing primarily on grows in that Western North American area. Okay, just a couple of the general characteristics of clovers. They are all herbs, either annual or perennial. They have usually 3 leaflets per leaf, which is where we get that common perception of clovers. And their inflorescences are head-like or umbellate with many flowers. Then I'll take a minute to just give a couple examples of the cool diversity of clovers because I think they're neat. So at the top of the slide is Trifolium stellatum, the star clover, which gets its name from once the flowers go into fruit, the sepals kind of spread out, like you can see in that picture, forming a nice distinctive star shape. Then in the bottom left of the slide is Trifolium macrocephalum, which is also known as the big head clover due to its very large flowers, much larger than typical clover flowers. But my favorite thing about that species is that unlike many other clovers, this species usually has 5 or more leaflets per leaf. So not always 3 leaflets, which I think is cool. And then the last one I'll mention on this slide is Trifolium incarnatum on the bottom right, which I just think is very pretty to look at with that super vibrant red color of the flowers. Okay, then a brief overview of the history of the taxonomy of the genus. The first big treatment of Trifolium was a monograph done back in 1984 by Zohari and Heller. But since then, there's been more work done on the genus, and more recently, a study done by Ellison et al. In 2006 looked at the phylogeny of the genus. They used most of the species. It was 218 species that they used in their analysis, and they kind of restructured the classification of the genus, dividing it into 2 subgenera, one of which was further divided into 8 sections. And the portion of the phylogeny that I have on the slide here is one of these sections, section Involucrarium, which contains all the New World species of clovers. So this phylogenetic study helped a lot with resolving the relationships within the genus, but there are still some parts that aren't fully resolved yet, the biggest of which being this section right here. So if you can look, there's a pretty big polytomy on this part of the phylogeny, so there's still a lot of work that needs to be done with these New World species, in particular some of the Californian species. So what I decided to work on in my research was this group in the red box up there, known as the Trifolium gracilentum species complex. Okay, so in the most recent treatment that included this group of species back in 2012, there were 6 taxa recognized in it. It included 5 species, one of which was divided into 2 varieties. This is known as a species complex because they have a lot of morphological similarities, and as you could see in that phylogeny on the last slide, they're very closely related as well. They're all native to Western North America, with their distribution centering around California, but also extending a little bit north and south of that as well. Just a few examples of the morphological similarities within this group is that they all have similar colors of flowers, all generally pink or purple. They have similar pubescence traits. They're all annuals. Other similarities as well, but those are a few examples. And since they have a lot of similarities and their ranges overlap a lot, there's been lots of difficulties with identifying them in the past. There's been some mis-IDs, and there's also been some debate over the best way to classify these taxa. So a few examples of the issues that people have debated about in the past. One of the species, Trifolium bifidum, some people have split it into two varieties. Other people have just lumped it all under the one species name. Then two of the other species, the ones that I have the pictures of on this slide, Trifolium gracilentum and Trifolium palmeri, have sometimes been classified just under one species, Trifolium gracilentum, but split into two varieties. So my main objective was to determine the best delimitation of the taxa within this species complex. And I did this by conducting a morphological study from herbarium specimens. I used nearly 200 specimens from various herbaria, as well as new collections I made myself on a trip to the California Channel Islands. I took lots of measurements on them and came up with a set of 70 characters that I used in my analyses. So the first type of analysis I did was a principal component analysis, or PCA, which generally just takes all these different characters, all that variation, and condenses it down into a smaller number of axes so that it's easier to visualize it. I hope the color isn't too light on the screen, but if you can see in the top left area of this chart, there's one species that clusters out pretty cleanly, that is Trifolium oleganthum, but the rest of the taxa all just came out in one big long cluster, not a lot of distinction between those groups. So this analysis was helpful for supporting the recognition of Trifolium oleganthum, but to get a better idea of what's going on with the other groups, I did a cluster analysis as well, and this helped out a lot with that. So this is still based on the morphological data. It's not a phylogeny, but again, it clusters the specimens out based on how similar they are. So I'll be going through each group on this chart to talk a little bit about the characteristics of all of them. Starting with Trifolium oleganthum on the far left in the light blue, which was the group that came out pretty cleanly on the last slide as well. So this is an example of a Trifolium oleganthum specimen, and you can see the geographic range as well. Okay. Then a couple of the traits that are useful for identifying this species is if you look at the margins of the stipules, with the arrow pointing to it right there, it has licineate stipule margins, meaning lots of pointed lobes on the stipules. And another easy way to distinguish this one from the rest of the group is by the presence of an involucre below the inflorescence, which is not found anywhere else in this species complex. Also, some of the floral traits, there's a lot of differences in the sizes of those features. A couple examples, when you look at the banner petal, it's a lot narrower than the banner petals in the rest of the species complex, and the claw length of the banner petal is a lot longer. And then one visual representation of that, this box plot here, you can see the banner petal claw length is a lot longer in Trifolium oleganthum compared to all the other groups. There's no overlap in that. Overall, this result didn't really surprise me. Just looking at the different specimens, it was clear that Trifolium oleganthum is very different from the rest of the group. And honestly, when I looked at all the literature about these different species, most of the time Trifolium oleganthum was not suspected to be closely related to the rest. Really, the first time that it was ever suggested that it would be closely related was in this phylogenetic study here, where it came out as sister to Trifolium bifidum. So that was kind of an interesting question for me as to how this species is so genetically similar to the rest of the group when morphologically it's very, very different. Okay, and then the next group I'm talking about is Trifolium ciliolatum in the green at the bottom of this chart. So this is also known as the foothill clover. If you look at the specimen, a couple of the traits useful for distinguishing this one is when you look at the shapes of the leaflets, the apices are usually much more rounded, but the easiest way to tell it apart is by looking at the calyx. So each sepal has this very wavy margin that is also, it has a lot of pubescence, a lot of hairs on it as well. So that's very distinctive to this species. So that's a pretty clear-cut way to tell this one apart. Next up is Trifolium bifidum, which is the one that has most recently been divided into two varieties. So I'll first be talking about the species as a whole, then looking into each of those varieties. So overall, Trifolium bifidum can be identified by its pubescence. It usually has a moderate amount of hairs on the leaflet midribs and also on some of the parts of the inflorescence that aren't usually hairy or not as hairy in some of the other species. And also if you look at the sepals, they're usually a lot more narrower than some of the other taxa. Okay, then going a little deeper into this one, looking at the two potential varieties. On the left is variety bifidum, and on the right, variety decipiens. The way that these have traditionally been distinguished is by the shapes of the leaflets. You can see on the left side, variety bifidum has this really deep indentation at the apex of the leaf, whereas variety decipiens has a much shallower notch there, which I did see in my results, but when I looked at that in the context of the rest of my data, this shape of the leaflets was really the only character I found that was different between these two groups. Everything else was basically the same, besides just normal small levels of variation. So I also looked at a histogram of this trait, looking at the size of that indentation at the end of the leaflet, and really it just looks like a continuous distribution, just a very skewed one, but you don't really see two peaks in it. So this suggests to me that it's probably just one group rather than two separate varieties. Likewise, when I look at the geographic distribution, the ranges do overlap a lot, though variety bifidum tends to be concentrated more in the southern end of the range, which suggests to me that maybe it's just a clinal gradient in this trait rather than two discrete entities. Then looking back at where this species is on the cluster analysis, that's in the black and red down here. Even though the two potential varieties do mostly cluster out pretty cleanly, if you look at the height of these branches, it's a very low height, again suggesting that there's not that much of a difference between them. So personally, I didn't think that the data supported them being continued to be recognized as separate varieties. Okay, and then the last two groups I looked at were Trifolium palmeri and Trifolium gracilentum. There's going to be a lot more to unpack here, but I will get to this. First up is Trifolium palmeri, the southern island clover, which is only found on islands. It's found on the California Channel Islands and Guadalupe Island off the coast of Mexico. Some of the traits that help with identifying this one are the shapes of the stipules. So the apex of the stipules is very long attenuate. It comes to a very narrow point that's a much smaller angle than in some of the other taxa. And also looking at the shapes of the leaflets, the apices of the leaflets come to a nice acute point. Then on the other hand, Trifolium gracilentum, the pinpoint clover, is seen here. It has a much wider range than Trifolium palmeri. The shapes of the leaflets are very different, in contrast to the very narrow, pointed leaflets of Trifolium palmeri. This species tends to have more obcordate shaped leaflets, and the stipules are different shapes as well. The apices of the stipules are usually a bit broader. Okay, then since previously these two species have sometimes been classified as two varieties within one species, I looked at these a little bit closer and performed a PCA just on these two groups. This time without all the other taxa in there, it is a bit clearer to see them coming out as two separate clusters, which matches up with the rest of the data since just looking at the two taxa, there are some very obvious differences, especially with regards to the leaflet shape that I mentioned. And there's also several other differences as well, like how much the stems branch or the width of the stem, as well as some other things. So there's a whole set of characters that help distinguish between these, not just one character. So this, to me, suggested that they should continue to be recognized as separate species. But the biggest question that my research showed is why Trifolium gracilentum was split into these two groups here. This was a big surprise to me. Nothing I saw in the literature suggested that there might be two taxa hiding within this species. So I took a closer look at this. I called this group on the left Group 1 and on the right Group 2. I did a cluster analysis just within all of these Trifolium gracilentum specimens, and once again, it came out on these two completely separate clusters. So when I looked at the data to see what could be causing this, I found that the difference between these two groups is flower size. The specimens in Group 2 tend to have larger flowers than those in Group 1. But besides that, I didn't find any other differences, and since clover flowers are so small generally and the floral parts are very small and you can really only get measurements on each of those parts by dissecting the flowers, that's not a very noticeable character to see, so it probably wouldn't have been noticed without a very detailed look at them like this. So I tried to find other ways that they could be distinguished besides these very hard to miss floral characters. Oh, well, oops, I forgot a slide. Sorry. I looked more in depth at some of these characters. These are a couple examples of floral characters that I measured, looking at the difference in size between the two groups. Like I mentioned, they tend to be larger in Group 2 versus Group 1. But what I thought was most interesting was when I put these on a histogram, this time I did see two peaks in the distribution, which suggests to me that these might be two discrete entities rather than just two ends of a continuous spectrum. So I looked at the geographic distribution to see if there was anything in here that could help as well, but the ranges of these two groups almost exactly overlap. I checked the elevation too. That's also basically the same between the two groups. So it seems like there's definitely something going on within this species where there may be two different taxa that should be recognized in there, but I don't quite yet have an answer to a firm answer on what these would be. I think it's something that would need to be looked at more in the future. So in conclusion, I decided to recognize 5 taxa. I recombined the two varieties of Trifolium bifidum just under the species name. There's obviously a big caveat on Trifolium gracilentum because I'm going to have to do more work on that and see what's going on in that group. And another thing that I think would be interesting to look at in the future is why Trifolium oleganthum came out so close to this group on the phylogenetic study done about a decade and a half ago when it's so morphologically distinct. So that's something else that I think would be interesting to test in the future. And now I will take any questions.